And Miyoshi Myopathy is a type of muscular dystrophy that specifically targets the distal parts of the legs, especially the calf muscles. And there are clinical data reported that the pathogenic dysferlin mutations can affect the protein expression level in the muscle cells. This is characterized by the decreased level or even absence of dysferlin expression in the muscle cells. So in dysferlin deficient cells, there is a defective function for the membrane repair, which is believed to lead to muscular dystrophy diseases, including Miyoshi myopathy.
Well, hello there. Welcome back to another episode of Rare Disease Podcast. Rare Disease Review is a not-for-profit medical journal organized by students from University of Toronto, University of McMaster and Western University. This podcast is created by the branch of Western University. The podcast aims to advocate for rare disease awareness and promote student involvement in the rare disease community. We appreciate your listening and support today. For further information on scientific translation of rare disease, please visit www.rarediseasereview.ca. The content of this podcast is not intended to be a substitute for professional medical advice, diagnosis, or treatment. So please always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay in seeking it because of something you have heard from this podcast. My name is Song, and you're listening to Rare Disease Podcast. For this episode, we're taking a look at a disease called Miyoshi myopathy. Miyoshi myopathy is a rare disease caused by mutations in the DYSF gene. It is a type of muscular dystrophy disease characterized by muscle weakness and atrophy, which means the wasting of muscle structure. Miyoshi myopathy first began in young adulthood on an average 20 years of age and includes weakness and atrophy of calves, leading to inability to jump, to run, or to walk on tiptoes. It develops over years with weakness and atrophy spreading to the thighs and gluteal muscles. The forearms may become mildly atrophic with a decrease in grip strength. The typical medical exam has blood exams showing an elevation of creatine kinase, often 10 times to 100 times above the normal values. Miyoshi myopathy is an autosomal recessive disorder that is heritable and is a part of the group disease known as dysferlinopathies, which are caused by the DYSF pathogenic variants. The management is usually physical therapies with mechanical aids, surgical intervention for orthopedic complications, respiratory aids, and social and emotional supports. To better understand the scientific research on Miyoshi myopathy, I spoke with Dr. Wang, a PhD graduate from the Biochemistry Department of Western University. Dr. Wang has dedicated her PhD studies to advancing the scientific understanding of the biomedical mechanism of the Miyoshi myopathy disease by studying its key player, dysferlin, a protein that is linked with skeletal muscle repair. Her work has made an amazing contribution to scientific research, and we're honored to have her here on our podcast today. So, welcome to the show, Dr. Wang.
Hello, thanks for having me. I'm really looking forward to the discussion today.
Yeah, it's so nice to have you here. We're really happy to learn about the disease from you. So, start off, can you tell us a little bit about yourself?
Yeah, of course. I finished my PhD degree in biochemistry from Western University early last year. I'm now a science writer in Toronto, and my research project over my PhD focused on the study of a protein that causes a series of muscular dystrophies. The protein's name is dysferlin, and one of the diseases that caused by dysferlin is Miyoshi myopathy, which is the disease we're going to talk about today.
That sounds great. So before we dive into your scientific research on Miyoshi myopathy, can you refresh our memory about the general knowledge of the disease?
Yeah. Well, I think you have given a nice and comprehensive introduction of Miyoshi myopathy. Like you said, Miyoshi myopathy is a type of muscular dystrophy diseases. And muscular dystrophy is a group of diseases that cause increasing weakness and gradual wasting of muscles in different body areas, from shoulders and arms to hips, legs, and feet. And Miyoshi myopathy is a type of muscular dystrophy that specifically targets the distal parts of the legs, especially the calf muscles. So when the disease first occurs, this happens typically in the adulthood of patients, 20 years old on average. And patients find themselves having trouble with standing on tiptoes, standing on heels, and climbing stairs. And over a period of years, the weakness and atrophy will spread to other body areas, such as thighs and gluteal muscles. And sometimes the forearms will also be affected to be mildly atrophic with a decrease in the grip strength.
Thank you so much for the really great information. So, what do you think is the cause of Miyoshi myopathy?
It is now believed that Miyoshi myopathy is caused by mutation in a gene named DYSF. And the DYSF gene is known to encode a single protein whose name is dysferlin. And dysferlin is actually a protein that can cause a group of muscular dystrophy disease. And this group of diseases together are termed as dysferlinopathy. And the Miyoshi myopathy belongs to this group. So it's a genetic disorder of a single gene.
That's interesting. So, as a genetic disorder, is there any available treatment for Miyoshi myopathy disease?
Since Miyoshi myopathy is a relatively newly identified disease, I believe it was discovered in the late 80s or early 90s. So the studies of the disease mechanisms is still not mature enough to find a cure or definitive treatment for the disease. However, there is some management that aim to prolong survival and improve quality of life for patients, such as physical therapy, like exercise, and depending on the extent of patient's mobility, patients can also use different mobility aids such as canes, walkers, and wheelchairs.
That's really interesting. It's good to know that patients are receiving physical therapy to manage to improve their quality of life. But I want to pivot the direction of the conversation to learning more about the mechanism of this disease. So you mentioned that dysferlin is the protein that causes Miyoshi myopathy. Can you give us an introduction to this protein?
Yes, of course. So dysferlin is a protein that was identified in 1998 to be the cause of two forms of inherited muscular dystrophy. And one of the muscular dystrophies caused by dysferlin is Miyoshi myopathy. And dysferlin is mainly expressed in our skeletal muscle cells. And in the early 2000s, some researchers made the fundamental discovery that dysferlin played an important role in calcium-dependent muscle cell membrane repair. They showed that in mice, so if the mice is dysferlin deficient, they will develop progressive muscular dystrophy compared to the normal muscle cells. And in normal muscle cells, they found the accumulation of dysferlin protein in the membrane patches after the membrane repair. And to date, more than 100 pathogenic mutations have been identified throughout the entire dysferlin gene. So dysferlin is a really large protein. It has over 2,000 amino acids. So it's probably one of the largest proteins in our body. And so these mutations are identified throughout the whole protein. And that was the data from almost 10 years ago. I believe there are more and more mutations being identified over the past years. And there are clinical data reported that the pathogenic dysferlin mutations can affect the protein expression level in the muscle cells. And this is characterized by the decreased level or even absence of dysferlin expression in the muscle cells. So in dysferlin deficient cells, there is a defective function for the membrane repair, which is believed to lead to muscular dystrophy diseases, including Miyoshi myopathy.
So it sounds like dysferlin plays a key role in repairing. Is that?
Yeah.
I'm wondering if you could tell us more about the mechanism of the membrane repair.
Yeah, of course. So as we know, the cell membrane is a very important part of our cells. So the cell membrane separates the interior of the cells from the outside environment to protect our cells against other stressors or and helps maintain the well-being of the cells. However, unlike plant cells or bacteria cells, our cells don't have are not protected by a hardened impermeable cell wall. So the loss of the cell wall leads to an unprotected cell membrane, which is more vulnerable to mechanical and chemical stress. And consequently, our cell membrane disruption occurs very frequently, especially in mechanically active tissues such as skeletal muscles or cardiac muscles and skin. Because of the cell membrane disruption occurs very frequently, our cells actually have evolved a membrane repair mechanism to quickly and effectively reseal the membrane breaches caused by the mechanical stress. And the wounded cells is able to survive only when the membrane repair system works properly. And to do the job of membrane repair, there are several components. And the first is the calcium in the cell, which is a messenger to activate the whole process by interacting with particular protein. And the major protein that calcium interacts is the dysferlin protein. So dysferlin can be recognized by calcium, and binding to calcium will activate the function of dysferlin. And after dysferlin is activated, it works together with a bunch of other proteins. And these proteins can bring or transport the lipid molecules, which is the component of membrane, to the disruption site and reseal the membrane. So that's basically how memory repair mechanism works.
Thank you so much for sharing all this information. I'm learning so much. It sounds like dysferlin is such an important protein in protecting our cells. And I know that your PhD research is focusing on this protein. So would you mind sharing a little bit more insight into your PhD research?
Yeah, so my research can mainly be divided into two parts. First part of my work is to elucidate how dysferlin interacts with calcium by looking at the change of the protein's 3D structure. And as we know, each protein has its unique 3D structure, and its structure is directly related to the function of the protein. So the structure change of dysferlin caused by binding to calcium is actually the determining factor for dysferlin to perform its function. So to understand the mechanism of how memory repair works, it is very important to know how dysferlin changes its function upon calcium binding and how it can interact with other proteins. So in this part of my research, I found that dysferlin is very sensitive to the change of the level of calcium in the cell. And because membrane disruption can cause an influx of calcium from outside the cell to the inside of the cell. And this influx is the reason to trigger the function of dysferlin and initiate the whole cell membrane repair process. And so this is the basic mechanism study for the function of the protein. And the second part of my research focused on the pathogenic mutations of dysferlin. So as I said, currently there are more than 100 mutations in identified dysferlin protein, and I picked a few of them, and I made the mutation of the protein and look at how these mutations change the structure of the protein itself and change how the protein interact with calcium. And what I found is that most of the mutations that lead to muscular dystrophy disease, including Miyoshi Myopathy. So these mutations, they can change the structure of the protein, leading the protein to misfold. Misfolding means that the protein cannot fold properly. So misfolding of the protein can normally cause aggregation of the protein and degradation by other proteins and degradation in the cell. So these mutations can cause the degradation of dysferlin protein. And that might be the reason why researchers can be found decreased level or absence of the protein expression in the patients.
Amazing information and wonderful work. I'm really grateful to have a scholar like you to contribute to the scientific society to advance our study. And it sounds like we are just steps and steps closer in understanding the disease better and having a treatment, which is probably our final goal. So for the future research direction towards the treatment of Miyoshi myopathy strategies, what do you think these directions would be?
So as I said, there is currently no cure for the disease, but there are many preliminary or even clinical studies going on by different research groups. So one interesting one using a short version of dysferlin. As I mentioned, dysferlin is a large protein, and using the full length protein as a drug is relatively more difficult for the patient to absorb. So the researchers, they designed dysferlin-like molecule, which is a short version of dysferlin. So they call it nanodysferlin. So they injected nanodysferlin into the muscle of mice, and they showed some really promising data. They showed that mice who receive this nanodysferlin injection can, at some level, restore the muscle functions. So which is pretty amazing and promising. And there are also some other strategies. The other two researchers, I mainly focused on gene therapy. So one of them is called stop codon breakthrough therapy, and the other is called exon skipping therapy. So these two strategies are targeting the mutations on dysferlin that cause Miyoshi myopathy. So they aim to change them. So because of the stop codon mutation and the exon skipping mutation, the dysferlin protein cannot be expressed. And these two strategies, they aim to change that on the gene level, which means that they can use some chemical or biological methods to alter that mutation and to restore the expression of dysferlin. Dysferlin can be expressed in the cell and to form its cell membrane repair function. So these are the main path for the future treatment for Miyoshi myopathy. And there are also many organizations that are helping to spread the knowledge and to fund researchers to study the mechanism and the treatment. And one of them is Gene Foundation, which is, I believe, in Seattle. And the other is Muscular Dystrophy Canada, which is a nonprofit organization in Toronto. If you're interested in learning about the disease and the treatment and research, I encourage you to check these organizations to learn more.
Thank you so much for joining us on the podcast today, Dr. Wang. I think your research has benefited such a huge population in advancing our understanding in Miyoshi myopathy disease itself, and also understanding protein, understanding genetic disorder. And we really appreciate researchers like you, contributing your time, your professional knowledge into studying this area and helping patients. So thank you so much for sharing that with us today. Hopefully we get to talk again in the future.
It's a pleasure to talk to you too.